The overall goal of this sequential fumigation incubation procedure is to estimate labile carbon and the potential carbon turnover rate in soil.
This method can answer key things in the soil microbiology field, such as the amount of substrate that soil microbes use to transform nutrients in the soil. One of the main advantages of this technique is that it's relatively simple to use and inexpensive.
Prior to collection, identify differences in soil properties and vegetation within the collection site. Using the appropriate coefficients of variation, determine the number of samples needed and the distance of variability. For heterogeneous soil conditions, designate a grid sample pattern wherein the distance between sample points on each transect is smaller than the distance of variability within an experimental unit. Clear organic matter from the soil surface at the sample points. Using an auger, collect each soil sample. For each transect of the grid, mix the samples in a single container. Place each composite sample in a cooler with ice packs immediately after collection. Once field work is completed, sieve the soil samples. Clean the mesh with water after sieving each sample. For each composite soil sample, place 3 individual 100-gram samples into beakers. Cover the beakers with laboratory film and pre-incubate the beakers at 25 degrees Celsius for 10 days. Then, for each sample, remove the film and place 1 gram of soil in a pre-weighed aluminum weigh boat. Record the combined weight of the moist soil and weigh boat.